What are species, and where do they come from? Darwin called this the mystery of mysteries, and named his great book On the Origin of Species. But an irony of that title is that in fact the origin hardly discusses the problem of getting two species out of one. The book is much more focused on convincing us that natural selection can in principle explain both the amazing diversity of the living world and its adaptedness, as shown, for example, in these three Hawaiian Drosophilus species. Species are real things, though it can often be impossible to say with confidence whether two related populations should be considered one species or two. And this simultaneous reality and difficulty is a major theme of this lecture. Biologists currently use three related species definitions or criteria or concepts when discussing the issues surrounding the reality and definition of species. Biological species are reproductively isolated populations. Because they never or rarely exchange genes, they have independent evolutionary fates. Phylogenetic or genealogical species are populations whose gene trees are always or predominantly monophyletic, which is to say, they're united by exclusively shared common ancestors. For this condition to hold, there must have been little or no gene flow for many generations. We can determine that they're monophyletic or close to monophyletic groups by doing research in molecular population genetics after the fact to see what their recent population history has looked like, but this is therefore a retrospective species concept. And indeed, we'll see that there's a deeply fundamental retrospective element to the definition of species, and that's part of the answer to the conundrum that species are real, but often very hard to define. Finally, morphological or taxonomic species are those that can be distinguished by consistent visible differences in morphology. This is the criterion that can be applied to fossils, and for the most part, the only one that can be, except for very recent fossils from which we might be able to get DNA. So let's apply those different species criteria to this case of the number of elephants in the world. It used to be thought there were just two, the Asian elephant and the African elephant. But then people started noticing that African elephants that live in forests, like this guy here in the middle, differ slightly in form from elephants that live in savannas. Subsequent population genetic evidence showed that indeed forest and savanna elephants are distinct populations that have experienced little gene flow for some time. And that's shown in cartoon form here in this little phylogeny, with the Asian elephants forming a cluster of tips that are monophyletic and distinct from all of the African elephants, who in turn form two distinct clades that are easily defined phylogenetically. So that's good. They're phylogenetic species, they're biological species, and once we learned to spot the differences, they're morphological species. Very simple. But many species are real, but completely cryptic. That is, impossible to tell apart by looking at them. For example, this copepod, Eurytomora affinis, is very abundant. It occurs in coastal marine waters all over the world. Carol Lee sampled many populations from locations all around the northern hemisphere and did DNA-level population genetics on them. Two genes suggested that there might be something like 8 phylogenetic species, and a feeling for that is given by this cladogram of populations from different collecting localities. Lee then went on to test the hypothesis suggested by her molecular phylogenies by crossing individuals from different parts of the tree. They're very tiny, they grow happily in the lab, so there was a great system for doing that. And in most cases, she found, they did not produce fertile offspring. They would produce offspring often, although not always. But in most cases where they did produce offspring, the offspring themselves were not fertile. So that seemed to demonstrate that the phylogenetic species, as seen from DNA evidence, are also biological species in the rather strong sense that they really can't, in most cases, successfully reproduce with each other, even though no one, not even an expert like Lee, can tell them apart morphologically. So what is the mechanism of speciation? How does one species become two? The easiest way to get reproductive isolation is simply to divide a population into two pieces, to physical populations that have difficulty exchanging genes through migration. This is the standard allopatric model of speciation. Allopatric is another one of these Greco-Roman made-up scientific jargon terms, but a very useful one. It means other country. Allo, yes, and patric, as in patros and patriotic. Okay, so the other country. And so here are the three steps, which I've cartooned on the right. We imagine a single interbreeding population living in a large range that has gradients of habitat conditions from one end to the other, say. So here I've imagined that a plus phenotype is favored at the upper left-hand end, and a minus phenotype is favored, at least somewhat, at the lower right-hand end. But gene flow, which, remember, is a strong, fast evolutionary process, keeps all parts of the species range fairly uniform and polymorphic everywhere. Then, step 2, a geographic barrier to migration appears. For example, a desert or a mountain range, reducing gene flow and allowing these populations, which have now become isolated, to adapt to their local conditions. The jargon term vicariance is often applied to events like this that create separated populations due to external factors, not anything that the organisms themselves decided to do. So this is a vicariance event. Splits our original population number 1 in 2, and then what happens? Well, just as in that two-population migration model, migration and selection model, that we looked at in a previous lecture, with no migration between these two pieces, they are free to fix for the genotypes that are more adaptive in the upper left-hand plus favoring part of the range, and the alternative genotypes that can now fix in the lower right-hand minus favoring part of the range. So geographic isolation brought about by this vicariance event of the appearance of a desert or something in the middle leads to genetic divergence between these separated portions of the population. If that remains the case long enough, then genetic and phenotypic divergences of various kinds can arise, enough to prevent gene flow even if the barrier to migration is removed, as it is here in step 3, and the populations come back together. If they are now sufficiently different in behavior, structure, whatnot, then they may be able to coexist ecologically in this zone that they've now reoccupied because it's no longer a desert, say. And if they're not inclined to mate with each other or can't mate with each other, then they will remain geographically isolated, and we have a proof that speciation is complete. However, this unwillingness to mate, or unwillingness or inability to mate and make hybrid offspring that are themselves fertile and that can cause genes to flow again from one end of the range to another, is not a requirement for them to be separate species. It's just, if it happens, a proof that they are, as is the case, of course, for many, many distantly related species alive at present. So here's an example, fairly close to us, a big one, caused by a vicariance event between the Atlantic and Pacific Oceans, caused by the emergence of the Isthmus of Panama. It started to appear in the Miocene as a ridge in the crust below sea level, caused by the South American plate crashing into the bottom of the North American plate. And as you know, that led to the appearance of what we now call Central America. By 5 million years or so, ocean circulation had changed, and there were no longer currents passing from west to east. And then by 3 million years, there was a dry land bridge completely cutting off the Caribbean Atlantic side from the Pacific side. And as a result, many populations of marine organisms, like these snapping shrimp, that had been contiguous populations across future Panama for a long time, many millions of years before, were now separated and free to evolve in their own independent directions. And they did so, resulting in a situation today where there are many pairs and trios and more complicated sets of sister taxa in this genus Alpheus, for example, snapping shrimp, that are each other's closest relatives. Very similar in appearance and behavior, easy to identify, especially with molecular means, as each other's sibling species. But in many mating trials that have been conducted by researchers in Panama, it turns out that in some cases they still mate and make offspring perfectly normally, but many are not very attracted to each other and or not fully fertile. So are they phylogenetic species? Well, yes. Here's these mitochondrial trees that I think you've seen before show they certainly are. Are they biological species? Yes, they are genetically isolated and diverging from each other and have been doing so for 3 million years or more. Are they morphological species? Well, in some cases maybe, but in other cases you can't tell. So the three definitions may be, notice, quite out of sync with each other. Geographical isolation can also arise through dispersal and colonization, and this is especially interesting and important in volcanic archipelagos such as the Hawaiian Islands, where new islands appear every million years or so at one end of the chain. In Hawaii, the youngest are on the right, over the hotspot in the Earth's crust through which the magma pours, creating spectacular volcanoes, and the older islands are the remains of that process, gradually moving to the northwest and getting older and wearing down. Because the ages of the Hawaiian Islands are all known, it is possible to reconstruct the histories of many species groups, such as these Drosophila fruit flies, who arrived on the islands long ago, many million years ago, and have been speciating like crazy on the islands ever since. So here's just a little clade of closely related flies related to Drosophila heteronura, which is the species one of the species recognized on the Big Island of Hawaii. But it has relatives on the older islands, at least going back to Oahu. This phylogeny is supported by molecular evidence, and it maps perfectly onto the history of the islands. So the oldest species, the most divergent, that is the deepest node in the tree, is that that connects Drosophila hemispisa from Oahu, a relatively old island, with the others, which occur on Molokai, Maui, and Hawaii. And notice the branching order in the tree corresponds exactly to the ages of the islands, with the shallowest nodes being between populations of Heteroneura and Sylvestra that occur on the same island, just on different sides of it, on the Hilo and Kona sides. So evolutionists love this type of evidence because it is an amazing proof of evolution. The shape of the tree, which can be confirmed by molecular sequencing, is predicted by the geology of the Hawaiian archipelago. And of course, the reason we're bringing it up here is that it shows how the speciation happened. An ancestor of Hemispisa, we might not have called it Hemispisa at that time, was on Oahu. When Molokai appeared, some individuals must have got blown over to Molokai in a storm where they found a perfectly good place to live with no other Hemispisa there, and they turned into what we now call Drosophila differens. Then that happens again with ancestors of differens getting blown to Maui after it appears, and we have today Drosophila planitibia as a descendant from that process. And then a little less than 1 million years ago, when the Big Island first appears, some of these Maui individuals get blown over to Hawaii, and they do well there and proliferate and establish, in fact, already different species and distinct populations within those species that may themselves be on the way to forming separate species in the much larger and more diverse ecology of the Big Island. How long does it take for species to form? There's no definite answer to that. Sometimes it appears that they can emerge very quickly. Other times it seems to take forever. Some interesting cases are known and have been documented of sympatric speciation happening almost under our noses in ecological time, from populations moving into suddenly created novel habitats or onto different hosts. The most famous example is that of this fly, Ragolitis pomonella, called the apple maggot, that was natively, it's a North American species, on native North American hawthorns. When European immigrants arrived in New England and New York and began growing apple orchards, Pomonella discovered the apple as an alternative host. Some individuals moved onto it, developed a preference for it, and formed largely isolated populations of Ragolitis pomonella that prefer to breed and lay their eggs on apples as opposed to their original host tree, hawthorns, which may be growing nearby, right next to modern apple orchards. So right in the same area, just a stone's throw apart, there are today these very recently incipiently speciated strains of the apple maggot, which really should be called the hawthorn fly, but they've become the apple maggot. They're still totally interfertile. They look the same, but they have a preference for apple, and they go to it, they stay there, they breed on it, and so on. So they are on their way, should that situation persist long enough, to becoming a distinct species, at least by some definitions. In a similar way, pea aphids, a pest of peas and related agricultural crops, have formed incipient species on, for example, alfalfa and red clover, which are also, like peas, legumes. Here's a remarkable data set produced a number of years ago by Sarah Vaya, showing that populations of the pea aphid from alfalfa, here A, on the x-axis for alfalfa, have high fitness on alfalfa and miserably low fitness on clover, on red clover. While conversely, pea aphids from red clover are well adapted to it, have high fitness on it, when transferred to alfalfa, have equally miserable fitness on this other host. They're very closely related. Everyone would call them the same species, but they are well on their way into the first stages of what we would recognize as speciation, just caused by agricultural modifications of their environment. These examples both involve human agriculture creating situations that are a model for how speciation might happen in nature, but similar things have happened, and in very small amounts of time, geologically speaking, with, for example, three-spined sticklebacks, little freshwater fish, that have independently evolved these two different forms, so-called limnetic and benthic three-spined sticklebacks that have different behaviors, hang out in shallow water and deep water respectively, have different amounts of armor to protect them from different predators that attack them in these different parts of the freshwater lakes created when the glaciers melted on North America around 15,000 years ago, leaving behind lots of newly created glacial lakes in Alaska and northern Canada. And that's happened over and over again. It's an evolutionary, it's really what amounts to a speciation or incipient speciation scenario that's played out clearly over and over again wherever this happened. And these fish, which actually were originally marine, become adapted to fresh water and form these two different types of three-spined sticklebacks independently, and all that over the course of just a few thousand years. So the long and short of this messiness of the process of divergence and speciation is that I, at least, and I think most biologists agree, think that it's best not to get hung up trying to force species to be absolute metaphysical categories. They're not. They're like clouds. They're communities of genes. And it's just much saner to think of them that way and let go of the need to be able to decisively answer the question, when did they become separate species? They can remain real or good species, it's clear, even if they occasionally do exchange genes with other species. This happens all the time. For example, in many Western North American and Eastern North American species pairs of birds. And we know that because bird watchers spend so much time observing birds very closely that they have over the years spotted many places where there is occasional hybridization between, as I say, Eastern and Western North American bird species that everyone considers good species. They're distinct. You can identify them from the bird book very easily. They mostly don't interbreed, but there are places where occasionally they do, and the hybrid offspring are perfectly viable. So those examples like that show that it's not a requirement to be a species unless you insist for arbitrary reasons that it must be, but there's no scientific reason to think that species can't remain real with a certain amount of leakage of genes in and out. These towies illustrate that principle. It's a wonderfully worked out example from southern Mexico, where the rufous-sided towee and the collared towee that again are perfectly distinct, and you'll find pages on them. They have separate names: erythrophthalmus for the rufous-sided and oci for the collared towee. every birder, every ornithologist would agree these are perfectly good species. But there are places in Mexico, illustrated by red blobs in this map. There, for example, where hybrid populations have formed, and the distinctness of the species has locally broken down, and you have a hybrid population. But other places not far away, like these, farther to the southeast, have both oci, the collared towhee, as shown by the stippling, and the rufous-sided towhee, as shown by the orangey color, in the same habitats, but observing their species distinctness. In these places, they are, as it happens, not mating with each other and making hybrids, while not that far away, they have done so, and the distinction has disappeared. And here's a place where there are collared towhees in part of this habitat patch, and it's got a chunk of it where there's now a population of hybrid towhees. So which way will it go? Is this just a short-lived aberration, or is it the beginning of a movement that will eventually erase the distinction between, that is, to turn what were two distinct species back into one? No one knows. Stay tuned. We just have to find out. There are some very instructive speciation brain twisters posed by what are called ring species. These occur in several widely divergent animal taxa. Perhaps the best worked out and most famous of them is this one, involving the west coast salamander Ensatina eschholzii, which occurs on the west coast of North America from British Columbia down into Mexico. The feature that's of interest here is that it forms a ring around California's Central Valley, with gene flow between adjacent populations all around the ring, and no individuals in the valley itself, which gets so hot and dry in the summer that it's just unsuitable habitat for salamanders. Eschholzii is highly variable in color. The figure illustrates some of these distinctive color morphologies, including this one, A, which is very plain, and a bunch of these have got names, subspecies names, and in particular this plain one, which will figure in the story, is the Eschholzii subspecies of Ensatina eschholzii, so you would call it Ensatina eschholzii, and so on for the others. The color patterns actually are not important to the story, nor are the names. So what's important is that here in Southern California are two places, one in San Diego County, one in Santa Barbara County, where this southwestern form, Ensatina eschholzii, comes in contact, in fact is sympatric with, lives together with, in some areas anyway, these inland forms, which are black and white blotched, and does not interbreed with them, does not exchange genes, and the two of them don't exclude each other ecologically. So in these locations, the two very different forms with different names and so on behave as if they are two different species by all of our usual criteria. They are morphologically different. They are phylogenetically different, as illustrated by this gene tree of mitochondrial sequences, and they are not exchanging genes, right? So by all of our criteria, they are two species in both of these places. However, if you walk the ring, either clockwise or counterclockwise from either of these places, you go between populations that are clearly each other's closest relatives. You, in some places, pass between these named blotch pattern forms and so on, but as the mitochondrial genealogy suggests, and as nuclear genetics also shows, they are all either currently exchanging genes with their neighbors, or they have done so in the recent past. That's true everywhere around the ring, except in these places that are the ends of the ring, where suddenly we have a jump, a big gap in the mitochondrial genealogy or in allele frequencies at nuclear loci, indicating in agreement with the large difference in appearance between the salamanders living together, that actually they're two different species. So, what's the answer to the question? Is it two species or is it one? Looks like two species here if you're on the ground, but if you travel around the ring, you never come to a place where there is a jump or a gap like there is at these two spots. The answer is, well, whatever you like, you can make the call, or as I think, both answers are actually correct, which shows that the question in this context is actually not altogether well formed. So how did this situation come about? The hypothesis is, and it is supported by the gene tree over here on the right, as well as other evidence, that after the recent glaciations, Ensatina eschholzii expanded its range south from Canada and the Pacific Northwest down into what's now California, and when it came to the Central Valley, it forked, and one population expanded along the coast, the other expanded along the Sierra Nevada, and then they met each other again in Southern California, so that the geography actually mirrors time. That is to say, there's more evolutionary time separating the black and white blotched and coastal forms of the species in the south than there is between any of the adjacent populations anywhere along the ring, or for that matter, going along the coast up to Canada or down into Mexico. Or, as Yogi Berra has very sagely said, it ain't over till it's over. Now, Yogi was referring to a baseball game, which isn't over till it's actually completely by the rules over. But I think the saying applies, and for a similar reason, to speciation. And biologists and you and I would all do well to just accept that you can't tell what the outcome was, in Yogi's case of the baseball game, or in our case of the incipient speciation process, until we have it in the rearview mirror and it's already done. That's the big point of this part of the lecture. Here's a diagram I made a number of years ago in hopes of helping make this clear. You may or may not find it helpful. Doesn't matter, but for what it's worth, here it goes. We have time graphed from flowing from left to right by this arrow. Let's say this is the present here on the right, then this is sometime in the past, and the stipple-ogram is meant to represent the gene pool of the species at time slices, right? So we can slice vertically through this genogram, if you like, anywhere we like. That represents the population at a given time. And at many times, there are these gaps that are meant to denote places where parts of the species population is not currently actively trading genes with other parts, and but the fact that they appear and then go away as we go from left to right means, yes, there were these gaps with this bit being isolated for a while, but then they came back together, right, and that everything was healed. But then somewhere around here, they start diverging in different directions, and as it happens, somewhere around this time, a division appears that looks at the time for all the world like one of these little temporary splits, but it persists. It never comes back together. And in fact, the two parts of this original population become so diverged that we would call them two species. So looking back on it, we say, well, there was a phase one when there was one species, episodes of geographic isolation, temporarily interrupted gene flow, but there was little consequence, little genetic or phenotypic divergence among the populations resulted from that because those episodes were brief. Then to go to the opposite end today, let's say we have two species, right? This is not complicated. They're not going to share genes again. We can tell that because they are substantially diverged genetically and in structure and ecology, and we've made gene trees and they show that they haven't actually been trading genes since way back at this time. Okay, no problem. The problem is here in the middle, in phase two, where they started to, it turns out they actually had separated, but they still could have fused back together again, and it would have been just like these other times in the past when they started to separate, but then decided not to, as it were, fused back together, and so nothing much happened. So here was a time of small but growing divergence. But there was never any time in here, never any day, when even with infinite knowledge of the state of the population and the ecology and everything, you could have said, yeah, so today there's still one species, but tomorrow they're going to be another, right? Or you come back tomorrow and say, yep, now they're two different species, but yesterday they weren't. There is no place in here where you can rationally say that. You, even here at the end of phase two, you could say, well, looks like they're becoming two species, but I don't know. If it's the case that they still could interbreed and everything, there might as well be called one species or two. We don't care. Okay. But at this point, there comes a time, but after much time, when it's quite obvious and you can say with confidence that, well, they did, as it happens, speciate somewhere in here, right? But if you were on the ground in there, you wouldn't have been able to make a case that that's what's going to happen. So there was never any time in phase two when they distinctly became different species, no definite time that it happened. And so we are fools on this view, which I'm sympathetic to. We're fools to insist that there should be some definite date, some switch point where it stopped being one and became two. And what is hard to get used to is the idea that even though there is no such time when they stopped being one and became two, nonetheless, it is a reality that they did become two, and that's not a figment of our imagination. That's reality. And that's just the way it happens. It's a historical process that occurs gradually. And support for this view comes from the fact that we find populations today at every imaginable stage in this progression. So the evidence is abundant that this is how it happens. It is gradual. For example, here's Drosophila again. I don't think these are all Hawaiian Drosophila, but they're bunches of species pairs. Each point is a species pair. And what's plotted is genetic distance from nuclear genes on the x-axis. So this is, think of this as molecular clock time. It's our best estimate of the time of separation of these species pairs. Or species or subspecies pairs, if you like. Okay, and then on the y-axis is a measure of prezygotic isolation, which simply means the tendency of these taxa not to mate with each other, given the chance by being put together in a vial in the lab. All right, so prezygotic because before a zygote is formed. Behavioral isolation. Okay, and so the pattern is that the allopatric taxa, those that live in different countries, remember, and in different places, who don't run into each other on a daily basis, they just gradually become more disinclined to mate with each other with the passage of time. And the ones that are fairly recently diverged from each other tend not to avoid each other at a very high rate. Whereas those pairs of species that live in the same place, sympatric pairs, even if they're only recently diverged from each other, they tend to show very high levels of avoidance of mating. So why is that? Right? That's an interesting puzzle for you to ponder. Might make a good fourth midterm question. Who knows? So, our summary. Recently separated populations may become biological, phylogenetic, and morphological species at different times and at different rates. Most speciation is probably allopatric just because that's the easiest pattern, and vicariant events happen all the time due to migration, due to changes in the habitat, due to the formation of mountain ranges. All sorts of things happen to split populations into pieces that are not, as it happens, experiencing a lot of gene flow for a while, and that gives them a chance then to diverge from each other. As we just said, the physical separation leads to adaptive divergence, which will be driven by selection, and also allows genetic drift then to fix random mutations that could lead to reproductive or developmental incompatibilities later if the two populations come back together, in which case they will experience, in addition perhaps to pre-mating isolation, but perhaps not, but they may experience post-zygotic isolation in the form of hybrid inviability or sterility, which will help cement the speciation that got going while they were apart. But while speciation is happening, I have argued, you don't have to agree, but I think it's a view worth entertaining at least. There is simply no way to know, even in principle. Right? It's not a matter of lack of knowledge. There's nothing you can imagine you could know with, Doctor Who technology to let you understand all sorts of stuff. We have no way of determining today, unless you actually have a time machine and could go to the future to see what happened, which is cheating. You would have no way to know whether the incipient speciation that you can see before you will actually end up going to completion, because it hasn't happened yet, and it won't be over till it's over. But then when it is really. Okay, so that's the end of our tour of speciation. Fascinating subject, but we have to leave it here, and we'll come back for one last lecture on evolution and health. See you.